Does anyone do space research? Raise your hand. Yeah, right on. Great. Yes, right on. Yeah, okay, great. Well, let's hope to change that. There's maybe opportunity now to communicate in the future, to start getting involved in these questions a little bit. There's a lot of relevance with space, in particular your immune system and aging. In space, there is this phenomenon of accelerated aging, which I know y'all are pretty interested in asking that question here, right? So thinking about those questions and how we can utilize this as a model is really a unique way to do that. So my talk is going to be about units in space. And I manage the Omics Lab for Health and Human Performance at UCF. So does anyone know what Omics is? Omics is the understanding of looking at transcriptomics, genomics, metagenomics, trying to understand the molecular questions that these cells are making, what then produces the protein responses that are coming from these cells, or ultimately the immune response. So I manage the Omics Lab. We have a few fun things in the lab that we can do questions in terms of space. We have next generation sequencer. We have a flow cytometer, flow sorter. We take information from samples from different exposure parameters that are very relevant for space, and I'll get to that in a second. We do this in mouse models and human models, and we try to see what kind of questions are happening. Okay. So what are the stressors of space? So there are a few different HRP-defined risks, or Human Research Program-defined risks, which is the group that fundamentally looks at human research at NASA as a human research program. We have things like altered gravity fields. Altered gravity fields combine of things such as launch and landing, hypergravity that's experienced during launch and landing. There is microgravity, or zero G, the influence of not having any gravity, or zero gravity. We have lunar, Martian gravity. As our immune system on Earth, we have developed with gravity. But when we get into these unique environments, we do see differences starting to happen. So that's an interesting question in itself. A stressor of spaceflight is behavioral and physiological isolation, and how that can influence different stress responses, and then ultimately how that contributes to your immune response. Ionizing radiation. We all know radiation can influence our immune system. It can influence our molecular structure. We know that radiation causes damage to DNA. So we can study what happens when DNA is damaged and how that influences the immune response as well. In closed and hostile environments, things like Moon and Mars adaptation, Earth readaptation, that's another element of stress that our bodies undergo in response to the spaceflight environment. Elevated carbon dioxide, hypoxia, hypercapnic-like conditions, how that influences the immune system. Pharmaceuticals for long duration is an important risk factor that we have to consider. In long duration spaceflight missions, you don't have the luxury of having drugs that can last for a long period of time, so having that preparation in place before astronauts go on these long missions is really important. And emergency medicine, triage, having those doctors in place on long duration missions is a really important concern as well. And then circadian shifting and how that is so relevant to our physiology. Our immune response peaks at certain cells in the daytime versus at nighttime. Giving vaccines, giving drugs, certain things like that are going to have a better immune response at a certain time of day. Considering that is really important when we're thinking about these kinds of questions as well. In general, things that affect our immune system in space include things like I mentioned, DNA damage, and radiation can cause that, and then we have certain responses that are triggered because of that damage. There's leukocyte imbalance. Different types of immune cells are upregulated at a certain time of day. And in particular, when we're talking about certain cells that we look at, I'll get into a little bit, granulocytes are one of those important cells that are really important for inflammation, driving inflammation as well. Rash formation. Rash formation is something that's seen in the spaceflight environment. This doesn't happen to all astronauts. This happens to astronauts that are predisposed to getting hypersensitivity reactions. Okay, so there is a genetic component that's linked to these things. Persistent mild inflammation is something that's also elevated in the spaceflight environment. This is all a factor in how long and the types of exposures that different astronauts are undergoing as well, but it is something that we start to see. Mitochondrial dysfunction. The mitochondria is what? The powerhouse of our cells, right? It's what drives a lot of questions with aging too, right? There's dysfunction in space with the mitochondria, okay? How we mobilize and make energy. Elevated heat shock proteins. Heat shock proteins are chaperone proteins. They're responsible for trying to repair cells when damage occurs. And that's something that we do see also in the space environment. Reactive radicals hand in hand with redox reaction, mitochondrial dysfunction as well. Okay, so lots of questions that we can ask, and lots of things that are relevant with the immune system. So previously, we wanted to sort of characterize some biomarkers that are really important in understanding what is happening in the immune profile in astronauts. And one of the things that we were able to identify was this elevated granulocyte to lymphocyte ratio and neutrophil to lymphocyte ratio. So the percentage of neutrophils and granulocytes in comparison to lymphocytes that are in blood circulation does increase with a period of time in human space. It also significantly increases when there is this high impact landing experience, this high stressful impact landing. We see this huge influx in blood of neutrophils to lymphocyte ratio. Okay. So that's giving us some clues that there is some inflammatory conditions that are happening in response to stress, acute stresses. I do include this one image here, which shows the personalized response of astronauts. Personalized response is a big question. We all have different immune responses to things. Monitoring that personalized response is a really important thing to do. We need technologies that are going to be able to do that type of monitoring. Grouping things based off of statistical significance is what we do generally in science, but we have to appreciate that personalized response in medicine. I put out recently a really nice report about the impact of microgravity on immunological states. It's a nice review article that some students that are under the program that I work on called the Aerospace Physiology Program at Embry-Riddle. They were able to put this nice review up. And if you're interested in understanding a little bit more about the different types of immune responses as a response to microgravity, please take a look at that, okay? So cosmic radiation being a big factor in how that influences our immune system. There are a few things that happen. We know that our immune system is very sensitive to radiation exposure. Immune cells have a fast turnover. They proliferate, but they need to proliferate very quickly. They have their own unique genetic profile. We're very interested when we think about those kinds of questions with genetics. So they're very sensitive to radiation-induced damage because of this ability to have a fast turnover. And ultimately, it is used a lot in cancer studies in order to eliminate the immune system to understand what is happening in different cancer studies as well. We know that there's different elements that are influenced by cosmic radiation. Chromosomal damage in lymphocytes in particular seems to be one of the bigger targets. They are the ones that are proliferating the fastest. And for example, a neutrophil is going to differentiate. Okay? You're going to have a certain type of cell that's going to be in your bone marrow. It's going to go through some proliferation events, but that ultimately is going to make a decision to be a certain cell. And once that cell then will leave that bone marrow and circulate in your body for a period of time, it may or may not become reactive to something, but then it ultimately will die. So each one of your immune cells have a different life cycle, right? A different lifetime. You can think of it, a different cell cycle. And so ultimately there's different sensitivities to those cells based off of the fact that they have these different life cycles, these different cell cycles. So that sort of just talking about that point, the general radiation influencing your immune system. Okay. We also can see that space radiation can inhibit angiogenesis. Do we know what angiogenesis is? Yes. Development of blood vessels, right? Making those blood vessels, making those ability for communication for blood to move through. Ultimately, space radiation can influence that process, okay? And that's kind of an interesting thing to consider when we're looking at fluidic dynamics in the microgravity environment. I know that probably addresses a few folks in the audience in terms of not just your blood vessels, but your lymphatic system also, okay? So your immune system's running through both of these systems. They're picking up stuff from your tissues, your lymphatics, and they're running through your blood system as well. Those types of systems are influenced in the microgravity environment. Asking questions on that is a very, very important question for NASA. We also know that the immune system in response to radiation can have influence on the immune system, but also on the nervous system. Okay. And this goes back down to this question of how inflammation drives that pathway into the central nervous system. Some stuff that we've seen in collaboration with glucose analysis, we do see that there's issues in spatial learning, cognitive issues in relationship to radiation-induced damage that affects blood-brain barrier permeability. Whether or not that is an influence of the monocytes attacking the blood-brain barrier or secondary to that attack is still a question of interest, but ultimately there are impacts on the central nervous system because of it. And Leonkis et al. In 2021 also found there was this increase in post-radiation exposure influences of creation of fatty acids. Fatty acids in circulation in high numbers can be inflammatory drivers. They can act as what we associate as danger-associated molecular patterns, and that can drive the immune response, ultimately the inflammatory response as well. And the fact that this persists for a long duration after site is an interesting question. We can study radiation now at Brookhaven National Labs. That's in Long Island. It is a unique, one-of-a-kind lab where we can study deep space radiation. So in terms of the radiation exposures that we are experiencing on Earth, they're very different from what astronauts experience on the ISS, which is also very different to what astronauts will experience going to the Moon and going to Mars. So we can study, in theory, the types of exposures that would be predicted to occur on a mission to the Moon and a mission to Mars. Now we can do that at Brookhaven National Labs. And we do that basically every year. So questions on radiation, tissues that we get back, please come talk to me, because there's an opportunity to ask these questions and questions that you may have, basically. I put this slide in because another thing that we do see in terms of radiation and combining this in a model on Earth with what we call simulated microgravity via Heinemann loading. Has everyone heard of Heinemann loading before? So Heinemann loading is the ability to tilt a mouse so that ultimately they're off their hind limbs. It's very similar to what we do with humans when we're talking about head down to bed rest. So the ability to move the fluid dynamics, that's very similar to what we experience in the spaceway environment. We can simulate those models on Earth, and we can study the immune response that happens to them. In general, the immune response is very different with various different types of radiation exposures. Going back to that same question of personalized medicine, going back to the same question of understanding what those specific triggers are. Okay. Any questions so far? None? Very recently too, I put this slide up because I just wanted to highlight one of my students who's now working at Cornell, doing her doctorate work, where we start to look into that question about how different stress hormones influence our immune system. And not necessarily just stress hormones, but hormones that are produced from our adrenal glands. Do we know hormones that are produced from our adrenal glands, by chance? Big hormone, really important for the stress response. I heard it. Cortisol, right? Corticosterone in mice, cortisol in humans. Ultimately, they come from the adrenals. And if there's damage to the adrenals, there's going to be issues with that production. The stress response and how hormones interact with the immune response is another really interesting question that I'm trying to get into a little bit more. And understanding what is causing that change, in particular things like elevated neutrophil to lymphocyte ratio. What is causing that movement issue with those cells? Is it something to do with this hormone change? Okay. So that's another thing I have to put that up. This is out right now. It's going to be part of a package that will be coming out this summer. but ultimately this is under nature scientific reports. And the other thing that we did see in this study is we do see very different sex-specific responses. Okay. So sex is a factor here, different sex-specific responses in the transcriptomic profiles of the adrenals and in the transcriptomic profiles of the blood. Okay? So again, characterizing these individual responses is very, very important. And I think we all understand that. What we're currently doing is to further address that question of leukocyte migration. Okay? And in response to different types of stress hormones, for example, looking at neutrophils in a microfluidic device, seeing how they move in response to inflammation, but also when they're moving in response to inflammation, also treated with cortisol. Okay, and seeing if there is changes in migration patterns. Okay. So these are just some nice videos that are giving some clues that with inflammation alone, we do see that there's this stickiness behavior that is happening with these cells, but when we start adding these additional hormones, they don't stick as well, okay? So their migration properties are a little bit different, and we're kind of delving into that a little bit more to see what those properties are. I think what would be interesting to this group, and this is why I put this up, is we're also developing a biosensor to monitor sweat, in particular looking at certain markers in sweat as a response to your circadian shifting. So one of the markers we're interested in is called orexin. Orexin is a neuropeptide. It's very much influencing our sleep-wake cycles, but it's also involved in inflammation. Why the question of circadian immunity is interesting is because of that response in general. We do have an elevated certain type of cell like neutrophils that peaks in the morning in response to things like elevated cortisol levels in the morning. And we want to see how in spaceflight, when we don't have that same circadian shift, that cycle is changed in spaceflight. It's like a 60-minute light-dark cycle that's experienced in spaceflight. You get different peaks of your immune system because of it, different production levels of cortisol because of it. And so we want to be able to track that more real time. Make these sensors, put them on astronauts, see what's happening with the stress level, see what's happening with other biomarkers of interest as well. So we're working on that project, and if you're interested in learning more, please let me know. Okay, and we are stuck on this slide. Okay, and just with that, I'd like to thank everyone that has been helping with these projects so far. If you do have any questions, I encourage you to please reach out to me. I have some cards available as well. And I do have this QR code up if you wanted to take an image of that as well. That links us to you to the Omics Lab, which again is what I manage out of at Embry-Riddle. And I have some stickers, I have some flyers for you if you're interested in the aerospace physiology program. I can talk to you more about that. And again, if you have any questions, please do so. Thank you for your time. Yes.
The little microfluidic, the cartridge?
Yep. Microfluidic device, yep.
Is that commercially available, or is that something you guys have?
Syn vivo.
What's that?
Syn vivo. Yeah, commercially available. Yeah. But we do have faculty here that may or may not talk to you more about the potential for creating microfluidic devices as well in-house. That's how we do it. Yeah.
Well, I'm not sure if this is directly related to the science part, but we have one college of hospitality and tourism. So one of the really talk about is some of the companies, commercial companies, they're really promoting space tourism. And actually, we have some which billionaires already went to the space and come back after 15, 20 minutes, right?
William Shatner.
Yeah. So my question is, with those companies have that goal in mind, and also based on the research on the immune system, what are the criteria that we need to set up if we want to achieve that goal and enroll it to the public or consumers so that we can pay at the same time to ensure its wellness and safety? Because the astronaut, obviously, they're very physically fit, but if we want to really create it as a commercial product, what are the things based on the finding that we need to see?
Yeah, I think that's a good and very important question. The rapid rate of how quickly these types of things are happening is very quick, and that needs to be much regulated in terms of the health impacts. In general, what we can inform folks like commercial aerospace and NASA communities is what we find in different duration type missions. So a 20-minute, 30-minute, 40-minute shot into a certain altitude won't have the same kind of immune response as a 3-year-long mission to Mars. There is that element of increased neutrophil and lymphocyte ratio, for example, with that launch and landing aspect, but that does recover, okay? So the question is then, what is the long-term implication of that? And if there is a long-term implication of that, and what is that risk per individual? And that's a question that does need to get further in, for sure. And I think we're working there. I think we're a little young to the game that's rapidly evolving. But it is an important question.
I had a question. So the fact that you mentioned about the stress response, I find fascinating. I don't know too much about the immune cells at this point. Oh, thank you. I don't know too much about the immune cells, but I've been wondering about the stress response in terms of pathogenesis and insulin linking to, I guess, glycolysis and that type of metabolic response. And I'm thinking more so in bone that you tend to get this replacement of tissue with fat tissue and adiposity. And my theory, for what it's worth, is that the body's trying to hold on to energy, and so it messes things up. And I wanted your thoughts on that, and also kind of linked to that, whether you think there's a role of the sphingolipids in there, sphingolipids. So for ionizing radiation, there's some drugs that I'm becoming aware of that are looking at ceramides, sphingolipid 1-phosphates, and I'm wondering, I'm guessing everything's involved, but I'm just wondering about how you see the puzzle in terms of pathogenesis, probably more so, rather than you, which I know is what you're looking at.
Yeah, Melanie, I wish I had that answer for you. That's a very complicated answer. I think we need to address the question of insulin. I think the gut is an important consideration with that, because it's all about how your gut tells your brain to make insulin with your pancreas. And then that is all developing based off of not just metabolic demand, but it's also because you're hungry. So there's a physical change. And how that is influenced in space is going to be another question as well. But definitely insulin, the receptors for insulin, the ability to make those kinds of connections are an important consideration. I think we need to delve into that question somewhere. We need to talk somewhere about that.
Yeah. I think it's just these lots and lots of thoughts coming around in my head, and I wonder.
One thing that we do see is that insulin growth factor is elevated in a space-like environment. So, saying that alone is telling you that there is demand for it, because it might not be getting the best signal for it. Evaluating that some more.
And another quick question, if I may. I think you may have answered this, but do you think of the cells in the peripheral bloodstream and the lymphocytes are a good kind of indicator of, I guess, space, for one of the better word, damage, rather than the monocytes? I know you mentioned those two, but
I have an obvious bias. I think, neutrophils are your first responders, so you're going to get that acute response. You're going to get it quickly, you're going to get it robustly. The fact that we're getting more than on Earth is an interesting question. And that came back to what I was talking about with Adam about, is this demarginalization question. And that's sort of what we're trying to understand with those microfluidic devices. Is there an element of making just more bone marrow cells and And do you want to make, in that certain stressful situation, to make more granulopoiesis is what it's called, right?
Yeah.
That's going into the blood system, or is it just that the ones that are in the blood system that are hanging out are actually going into the pool of blood circulation? And so that's sort of a question we're trying to address. It's a trickier question to address in vivo.
Yeah.
So we can kind of look more at those cell surface markers for what it's worth to see if there's elements there that